Hi guys, thanks for tuning in on my video abstract. My name is Frederik Engelen and I am a PhD student at Ghent University at the Faculty of Veterinary Medicine. I am currently involved in a research project funded by the federal government, during which I am determining the presence of enterohemorrhagic Escherichia coli on Flemish dairy cattle farms. Escherichia coli is a gram-negative rod-shaped bacterium belonging to the family of Enterobacteriaceae. The majority of E. coli are commensals in the gastrointestinal tract of humans and warm-blooded animals. However, some pathogenic strains have acquired the ability to cause severe disease. Shiga toxin-producing E. coli, or EHEC, is a group of E. coli which is characterized by the production of Shiga toxins. Enterohemorrhagic E. coli, or EHEC, is a highly pathogenic subset of EHEC. E. coli serotype 0157H7 is most frequently implicated in human disease. However, non-0157 serotypes have been increasingly reported as important foodborne pathogens. Besides 0157H7 EHEC, a number of non-0157 EHEC serotypes have also been linked to severe human disease, including 026, 0103, 0111, 0121, and 0145, which are referred to as the pathogenic gang of 6. The aim of this research was to determine the presence of EHEC in dairy cattle on Flemish farms. Cattle are the major natural reservoir hosts of EHEC, which harbor the bacteria in the gastrointestinal tract without clinical signs and subsequently excrete EHEC in their feces. Contamination of meat with feces or intestinal contents of cattle during slaughter, followed by consumption of undercooked meat, is considered the most common route of transmission. Other common outbreak-associated food vehicles include unpasteurized milk and other dairy products, fruit juices, and vegetables. Although adult cattle are unaffected by these organisms, some non-0157 serogroups have been linked to diarrheic syndromes in young calves. EHEC utilizes a combination of two virulence strategies to colonize the gastrointestinal tract and cause disease, namely the production of Shiga toxins encoded by the STX gene and the formation of attaching and effacing lesions encoded by the EAE gene. In 95% of all cases, a characteristic and usually self-limiting bloody diarrhea is observed. However, in 5% of the cases, Shiga toxins enter the systemic circulation and cause a variety of clinical symptoms known as the hemolytic uremic syndrome, including hemolytic anemia, thrombocytopenia, acute kidney failure, and encephalopathy. Individual pens were sampled with overshoes. This technique involves sampling the pen bedding by walking around in an H-shaped track using a pair of disposable, liquid-absorbing overshoes. Animals were roughly divided into 3 age categories. A total of 88 overshoe samples were collected from 19 Flemish dairy cattle farms. 9 samples were taken from newborn calves, 51 samples from young cattle, and 28 samples from adult cattle. 2 pairs of overshoes were taken per pen to investigate presence of both 0157 and non-0157 EHEC. These samples were enriched in modified tryptin soy broth supplemented with novobiacin or Brilla broth for 0157H7 EHEC detection and non-0157 EHEC detection respectively. After incubation, an 0157H7 specific immunomagnetic separation was performed, followed by plating onto CT SMAC. Presence of the 0157 serotype was confirmed by latex agglutination and subsequent serotyping. Non-0157 enrichment cultures were subjected to a selective acid treatment, followed by plating onto Chroma-Agar and Chroma-D EHEC agar. A pool of 10 suspected colonies was tested for presence of virulence genes by multiplex PCR. Individual colonies from pools testing positive for both STX and EAE were analyzed for presence of virulence genes and confirmed by a serogroup-specific multiplex PCR. 19 dairy cattle farms throughout Flanders were screened for EHEC presence. In total, 11 dairy cattle farms tested positive. 0157H7 EHEC was present on 5 farms amongst young cattle and on 1 farm amongst adult cattle. Non-0157 EHEC was present on 1 farm amongst newborn calves with 0145 on 6 farms amongst young cattle with O26 and O103, and on 4 farms amongst adult cattle with O26, O103, and O145. In samples collected from 8 farms, only the STX gene was detected. To conclude, we can state that EHEC is still frequently present on dairy cattle farms and tend to have a higher prevalence in young cattle. A number of samples containing only the STX gene were identified in this study. However, unlike O157H7 EHEC, there is currently no reliable and simple isolation method for the detection of non-O157 EHEC, possibly resulting in an underestimation of actual non-O157 EHEC infected samples.